Hello, science listeners. Science Made Fun delivers quality STEM activities for kids. Our podcasts are created to spark curiosity for both children and adults. What do we do at Science Made Fun? Our scientists come to your location and convert your classroom into a living laboratory. This fosters a lifelong love of science and makes the experience both meaningful and memorable for everyone. Feel free to follow us on Instagram and Facebook. Just search for Science Made Fun. Hello, science listeners. Today we're talking with Dr. Melinda Groser. She is a professor at the University of North Carolina, Asheville. Her current research is on nucleotide signaling, molecules, and Staphylococcus aureus. She is also going to talk a bit about the art in STEAM. Enjoy. Hello, I'm here today with Dr. Melinda Groser, who is a biology professor at University of North Carolina. Is there anything else that if you were to introduce yourself, how would you do it?
Microbiologist for work purposes, and then I also always include that I'm an avid fiction reader.
An avid fiction reader. That's awesome. What are you reading right now?
Well, I usually have about 3 books going at once. So I'm listening to an audiobook called Austen at Sea, which is Jane Austen adjacent. And then I am reading a book called Heart the Lover by Lily King. It's like a contemporary fiction. And then that's on my Kindle. And then I also have a paperback of Visit from the Goon Squad.
Oh, all of them are pretty good. How do you keep up with all of them at the same time?
They all have to be pretty different.
Yeah.
But I like to have one in each modality.
Gotcha.
So,
one audiobook.
Yeah.
One by the bed probably or whatever.
Yeah, I like my paperback, and then I like to have a Kindle one. That's, a little more flexible.
Were you always interested in science as a child?
As long ago as I can remember, yes.
Could you think of a specific incident that really got you fascinated as a child?
So I was very lucky. My dad is also a professor. Mm-hmm. He is a horticultural scientist at the University of Florida. So whenever I got to go to work with him, which was whenever I was sick from school, or if we had a day off school, I was in his lab a lot. I got to see what he was working on. And so I remember being very young and getting to look through a microscope and look at plant tissue that was fluorescent because it was expressing GFP, green fluorescent protein, which is a gene from a jellyfish. And I didn't actually connect until later that green fluorescent protein that I work with now was what I looked at then. He just told me it was the jellyfish gene. But I remember thinking that was really cool. And then I also, for science fair projects in like middle school, got to make hybrids of citrus plants.
Really?
Yeah, with like a little paintbrush in my yard. He had me cross-pollinating plants. And yeah, it was really fun. So I was very fortunate to get to do stuff like that as a child and be exposed to it really early on.
So are you specifically interested in plants? Because I know you study bacteria, right?
Yeah, bacteria are my main area of interest and expertise. My undergrad research was in plants, but I actually focused on mitochondria in plants, which is an organelle that evolved from bacteria. So it was a nice transition to working with bacteria from there. Yeah.
Could you think of anything specific that really fascinated that you learned something new?
Anytime, or like in my intro to science years?
In your intro to science years.
In my intro to science years.
That got you super excited.
Yeah. Let's see. When one of my science fair projects was very fun, I was rescuing embryos from like shriveled citrus seeds and trying to get them to grow. They wouldn't have grown on their own, but they grew by tissue culture. Mm-hmm. And that was cool. There's I guess I didn't discover it at the time, but some of those plants that I rescued, my dad is still using in experiments now.
Wow.
So that's really fun.
Awesome.
Yeah.
That's amazing. It's kind of like generational study, and maybe he will have a student or someone else to also study those plants.
Mm-hmm.
That's pretty awesome.
I get pictures of how they're doing from time to time. But yeah, as far as for microbiology, I first got interested in it after junior year of college. I did an internship in Switzerland for the summer, and that was my first time working with bacteria, with Vibrio cholera, which causes cholera. And I was looking at gene expression when cholera is growing on crustacean shells, and trying to understand when it's growing on crustacean shells, it becomes competent, meaning it can take up DNA from its environment.
Oh, wow.
And we were trying to understand the regulation, the genetic regulation of how that happens. And that kind of got me hooked on microbiology in particular.
But specifically, what are you researching right now?
Here, yes. What am I researching here? I am studying Staphylococcus aureus. I have a few different strains that are also MRSA strains, which stands for methicillin or multi-drug resistant Staphylococcus aureus. And we are investigating different mechanisms for how staph is resistant to antimicrobials produced by the immune system during an infection. We're specifically looking at a couple of signaling molecules that staph accumulates inside the cell, inside the cytoplasm during stress. And we're trying to sort of disrupt their normal signaling pathways. We're trying to create strains that make too much of these signaling molecules or don't make enough of them, and study the consequences in terms of how they respond to stress and how they
What do you mean by stress?
So your immune cells are trying to kill bacteria during an infection. So they're going to make chemicals that are antimicrobial, including hydrogen peroxides. If you've ever treated a wound with peroxide, kills bacteria. Your immune system makes similar chemicals. Another one's nitric oxide. It will make highly acidic compartments, and that can kill bacteria. So these are all different stresses like that your immune cells are imposing on bacteria, and we recreate them in the lab. Also antibiotics.
So that's how they're functioning.
Yeah.
They're producing these other chemicals.
Your immune cells are to try and kill bacteria, and we're trying to study how do bacteria survive those things, because they have to. If they're causing an infection, they can work around it and survive. And so our idea is the idea is not just mine, if we can understand how is staph able to resist all these different chemicals and identify specific proteins and genes needed to resist these chemicals, those represent good drug targets.
Okay.
We would be essentially, like, by targeting one of the genes or proteins we identify with a drug, we would be helping the immune system to kill staph.
So you know how to treat it better in the future.
Long term, yes.
Long term. Were you specifically interested in staph, or was that just a research assignment that came up?
A little of each. So when I started grad school, we got to rotate through 3 different labs, and we could pick the labs. And the first one I rotated in worked on staph, and that was the one that I enjoyed the most. So I stuck with working on staph for my whole PhD. And then I did something else for a little bit for my postdoc, but then came back to it because I was really interested in staph in particular.
Well, I would say that obviously it's super important, because staph infection is it's really bad.
Yes.
It's can be deadly.
Yes. It's one of the most drug-resistant pathogens that is causing significant public health costs with how prevalent it is in hospitals and now in community settings as well.
And you also create art with bacteria.
I do, yes.
What inspired you to do that?
So the American Society for Microbiology actually has a bacterial art competition at their conference every year. And so I knew about that, and I had seen some of the really cool creations people turn in. And so I actually started doing it first as an assignment in my microbiology class. We isolate, or the students isolate soil bacteria in that class, and a lot of them are naturally pigmented and are really colorful. And so I thought a fun assignment would be to have them make art at the end of the semester with their bacteria, and then I got into doing it as well to show them examples and just having fun with it.
So different bacteria create different colors as they grow.
They do, yes.
And I see you have a whole palette of colors. Could you show us one of those?
Yes. So this one, there's different colors. You can see they're maybe not quite as vibrant. These are all soil isolates that students in my classes have isolated from local soil. So we've got like a dark purple, we've got a red one, we've got some oranges and yellows and tans. So these are common colors.
How do they isolate them?
They will take soil samples, dilute them in water, and the microbes will be in the water. And then they make serial 1 to 10 dilutions and spread them on plates. And eventually you'll dilute them enough that you're spreading them to single cell thickness. And then each single cell will grow into a colony. And that colony will be a pure culture of
and you
whatever
can transfer that colony to your art.
Exactly.
To your bacteria art.
So like this dark purple one was a single colony on a plate, and we were able to pick it with a toothpick and amplify it further.
So, and I see you also have you have two that are like color palettes.
Yes.
But then you have one where you've written
I do have an art one. So this one, you can see they're a little bit more brightly colored.
They are bright.
These are all E. coli. It's a non-pathogenic commonly used lab strain of E. coli. And they have been transformed with a plasmid, so an extra circular piece of DNA that contains a gene encoding a chromoprotein, a colorful protein. So these are all genetically engineered E. coli. And that makes they're making colorful proteins.
Making colorful. And they do that so they're easier to study?
Some people use the color as an indicator that the plasmid is in the bacteria. If it turns the color, that plasmid is there. In this case, part of the goal is as an activity for students to transform E. coli, to genetically modify it and have a visible readout at the end. So I don't know that there's necessarily like a goal of what to do with the colorful E. coli, but it's a good exercise for students, getting a plasmid into bacteria and seeing the consequences of that.
They're really bright. I see a pink one, purple, blue, yellow, all the colors.
And these, the plasmids also contain an antibiotic resistance gene. So we grow these on plates that contain an antibiotic.
So you've probably created many, many of these already, right? What is an art piece that you created that you're pretty proud of, your bacteria art?
Well, I brought one to show you, just as an example. So this one just says UNCA biology on it.
Awesome. With some mushrooms.
Yeah. A mushroom and that's supposed to be an E. coli with
E. coli.
A flagella and a flower. So just an example, it's easy to use a toothpick or a pipette tip and write with the bacteria. The tricky part is you cannot see the outcome until it has a couple of days to grow in the incubator. So it's like writing with invisible ink, essentially, and then you have to wait a few days to see how it turns out.
And that usually takes how long?
E. coli will grow overnight, 24 hours. I usually leave them in at least 48 hours because the pigments are slower to develop. So 48 to 72. So yeah, they're fun. Some of the ones I'm proudest of have been book covers that I've tried to recreate.
Oh, wow. Which book cover?
One of my favorites is the cover of House of Mirth by Edith Wharton. That was a fun one to do.
That's a pretty good project. I think that's kind of fascinating. It's like you're interested in the book. Maybe students are also interested in a specific book, their favorite or whatever, and then they could reproduce that.
I did have some students in my class do book covers after that. Yeah. And I like to post them and write also a bacteria fact that something in the book made me think of too.
Oh, is metaphorically?
Sometimes yes, or sometimes more direct.
Referring to the bacteria.
Yeah.
How did you become a professor here, and what has your experience been so far?
Yeah, so I after grad school, I was in California for a few years doing postdoc research, and I really decided during that time I really wanted to teach undergraduates, and so I started teaching as a postdoc. I taught summer classes at UC Berkeley in microbiology and got some experience and was like, okay, this is what I like to do. And so after a couple years of that, I decided I was ready for faculty jobs, and I wanted to be back on the East Coast, be closer to family. So I specifically applied to jobs at smaller liberal arts colleges where there would be small classes and I could incorporate research into my teaching and do research with undergraduates specifically. So that put this school, they had a job opening in microbiology at the time. I also knew I really liked Asheville. I was at Chapel Hill for grad school, so this was our vacation spot. And so I really liked the area and interviewed at a few different places and ended up deciding to come here because it was a nice balance of what I was looking for, research with students and teaching in my subject area. And I've really enjoyed it so far. We have a great department. I like being in a general bio department where everyone does something completely different because I get to learn a lot from colleagues who do field work. I get to go out in the field with them.
And you collaborate with the other
research
professors as well.
Yes.
Could you tell us anything about a specific collaboration that you've had with professor?
I've had a couple. So we just started a new one with Dr. Camilla Filgaris, who is an entomologist in the department, and she is also an expert with nematodes. And so it might sound surprising, but other people have used C. elegans, which is a nematode, as an infection model, an animal infection model for staph infections. And nematodes will eat staph and then have a gut infection, essentially of bacteria. Oh. And you can study how virulent different strains of staph are by comparing how well nematodes survive and how well the bacteria replicate in the gut. And so Dr. Filgaris is helping us set up a model where we're infecting some nematodes with staph, and we're going to compare between our wild type strain of staph and some different mutant strains to see how well they cause infection in nematodes. And she's got a really cool new instrument upstairs. It's like a small organism flow cytometer. It sorts little tiny microscopic organisms like nematodes. And her idea is that it should be able to tell us if nematodes are alive or dead, like in high throughput. Nematodes can just go through the machine and it'll mark, was this one alive or dead, based on its infection status.
So it just identifies whether they're alive or dead, or that specifically identifies the nematode itself, like this variety or this.
It can do that as well, but so she uses it for that with soil organisms to identify it. We're going to be using it with all the same species of nematode.
Okay.
So it'll hopefully be able to tell us the outcome. We haven't done it yet, but we're in the process.
That's awesome. Where's your dad's lab located?
He's at the University of Florida, and he works at an off-campus research station, the Citrus Research and Education Center.
That's awesome. And I want to ask, how was it living in Sweden?
Switzerland. Yes. Switzerland. It was amazing. I was there for a summer, which was not long enough, but I was in Lausanne, Switzerland, which is in the French part. It's right on Lake Geneva.
Okay.
And it was beautiful. Amazing food, amazing cheese.
Amazing cheese.
Yeah. It was at the EPFL, which is like the Swiss Federal Institute of Technology, and also a really great research institution and really cool international environment to work in.
Other than your dad, who obviously was not just your dad, but kind of like a mentor in a lot of ways, what other do you have any other mentors currently, or anyone that has helped you on the path that you are now?
Yes, many people have. So I had a really great dissertation committee as a grad student. So my advisor was really supportive, my PhD advisor, and then I had a group of like 4 other faculty who all gave me lots of frequent advice and who I've stayed in touch with, many of them, not all of them, but many of them since grad school. So it was fun. Like when I got tenure here, I texted my PhD advisor right away, like, got tenure. And awesome. So that's been really, he's continued to write
He's very excited for you.
Yeah, he's continued to write me recommendation letters and to help. He's sent me some of these strains to help get my lab started here. And then I also have had mentors who had similar jobs as this. A lot of the people I worked directly with in grad school were at a really large research institution. So they were running big labs and not doing very much teaching. And so I kind of had to seek out mentors who were at smaller liberal arts schools doing the kind of job I wanted. And there were a few people who were like a couple years ahead of me in grad school, or who were postdocs while I was a grad student, who were really generous in sharing application materials and like teaching materials that they had used. And so that was also really, really helpful.
And now you yourself have become a mentor.
Yes.
To many other students?
Yeah, I have usually 5 or 6 students in my lab, sometimes more at any given time, undergraduate students. So trying to help get them started in whatever they want to do. And I've been a part of the peer mentoring program at UNCA to help newer faculty members kind of get on their feet. I had mentors through that program as well.
So that leads me to the very last question. If you were to give any advice to anyone wanting to get into research, microbiology research, what would you say? Like if you were talking to a teenager, for example.
Yes. I would say to find an area of research that you're really excited about, because there will inevitably be things that are really tedious about it. And so I think you have to find something that you're really genuinely interested in, like somewhat as a hobby as well as just your work. Otherwise, you might lose motivation. And I think you also just have to have persistence. Grad school is hard academically, but the hardest thing for me is that you just have to stick with it, even though sometimes things might not work for like more than a year at a time. And so I think the biggest like thing that determines if you're successful or not is, can you just stick with it even when everything fails for months on end? Then you can do it.
So stick with it. Keep that fascination fresh in your mind, and probably take some breaks sometimes, right?
Yes.
Give yourself some breaks.
Make sure
How do you
have
take
hobbies.
breaks?
Yeah. I have a lot of, probably too many hobbies.
But you go hiking, right?
I go hiking. I love to be outside with my dog. So we're walking and hiking. In grad school, the biggest way I took breaks, I lived across the street from a lake, and I had a little kayak, and I had wheels for it, and I would wheel it down the street to the lake and go fishing pretty much every weekend.
Nice.
So I do that, and then I read a lot. And then my other one is crocheting. Just furiously crocheting gets all the stress out.
All right.
Yeah.
Dr. Melinda Groser, thank you so very much.
Thank you.
Thank you. That concludes our episode with Dr. Melinda Groser. Dr. Groser, thank you so very much for your time and dedication to research. Are you ready to have us turn your classroom into a living laboratory? Visit sciencemadefun.net to find a location serving your area and book programming.